and I'd like to thank Norbert for inviting me. And I do work on olfaction, and it was really nice to hear Bertram's talk this morning, because basically he's using the piriform cortex as the way to stimulate the seizures. And so let me talk to you a little bit about what we've done in the lab and how that is related to what we're doing here. So basically, and I'm going to talk about work we did this year. So there is an involvement of olfaction in epilepsy. So it's well known that there's a subset of human temporal lobe epilepsies where there's complex partial seizures that are followed by olfactory hallucinations, olfactory aura. So that's afterwards. But there's also a thought about before. The auras appear to involve the piriform cortex, which is what we talked about. And there's a subset of patients that can often prevent the seizure by smelling an odor, which is an interesting thing. Now, so let me tell you about what we've done. And I'll start with a drawing from Cajal, from Ramon y Cajal. And this drawing is basically a drawing of the olfactory system. So these are the olfactory sensory neurons in your nose. And those express in humans about 1 of 250 receptors. In a mouse, it's about 1 of 1,000 receptors. And those send the signals from the olfactory sensory neurons all the way to the glomeruli. And there they go to tufted cells or to mitral cells. And those cells then send the signal out to the piriform cortex. Now, of course, these are all excitatory. This one to the mitral cells, and then the mitral cells over to the piriform. But there's, of course, inhibitory cells. The granule cells over here are inhibitories, and they send signals over here to the mitral cells. There's also some over here, the inhibitory neurons. And there's a large number of inhibitory neurons, GABAergic, over here in piriform. Okay, now, of course, the output, but of course that is pretty evident from the talks we've heard before, the output from the olfactory system does go over to places like the amygdala, of course, and entorhinal cortex, and others. Okay, so let me talk to you about some work that has been published in the past about showing that the olfactory system can start seizures. And so first I'm going to talk about work from Nguyen and Riva. And so this is basically what they did is they made a new mouse. And in this mouse, they took one of the olfactory receptors, it's called I7. And they expressed that in every single olfactory sensory neuron. So normally, a single olfactory sensory neuron will only express in a mouse 1 of the 1,000 receptors. In this case, basically almost every single one did. And you can see that here, basically. So this was also expressing GFP, and you can see in the glomeruli, so this is where the olfactory sensory neurons send their axons, that basically a large number of glomeruli were innervated by these olfactory sensory neurons. So the question with that was, what happens if you stimulate that one receptor, and what happens? Do you get seizures? And they did get quite a bit of seizures. So over here, they used octanol, and octanol happens to be the odor that stimulates this I7 receptor. And it elicited seizures quite a bit. They expressed either, well, they used either 1% or 10% of the octanol, and they had a large number of seizures in a subset of the mice. So that was interesting. So basically, in this case, it was basically 2 out of 7. In this case, it was a larger number. So the question is, what was happening? Now, in this case, you were stimulating the olfactory sensory neurons. So if you look at CFOS, which is what's shown here, you don't see basically a difference between those that show the seizures versus those that do not show the seizures. There's large difference with control. So this is the olfactory sensory neurons, and because we're looking at FOS, basically it's only a small number that respond in the control. Control is basically them in a cage. And there is a little bit of a response just because the cage smells. But then when you put the octanol, you get a large number of those cells to respond because they're all expressing I7. And this is not different from the strong seizure. So if you look at the strong seizure, basically the same thing. You get a large number of these cells. So where is the difference? Where are you getting the difference that will get you to seizures? And the difference happens first in the olfactory bulb and very clearly in cortex. So let me show you. So basically that's shown here. So this is basically the olfactory bulb, and the input is out here to this glomeruli, so that's where the axons from those olfactory sensory neurons that I showed in the last one are. And again, in color, you see FOS. So you see FOS expression. And this is piriform cortex. So basically, the axons from these mitral cells will then go back to piriform cortex. So in a control, you get a good number of these to respond just to the air in the cage, and you get a small number of cells in piriform cortex. In the cells that were expressing the I7, you had two totally different mice. You had the mice that had no seizure, and the mice with the exact same odor, they had a fairly strong seizure. And if you looked at FOS, the difference was large. Basically, a relatively small number of cells would respond here in the olfactory bulb. A relatively small number of cells would respond in the piriform cortex, and yet there was massive response both in olfactory bulb and piriform cortex in those that basically responded by seizures. So that's an interesting thing. So basically the system can actually turn this on. Now, what happens beyond the olfactory bulb and the olfactory receptor at the piriform cortex? So let me go back to the drawing. So basically this is the olfactory sensory neuron, then you have the olfactory bulb, and then you have piriform cortex. So first of all, it is well known that piriform cortex does participate in seizures. So this is work, for example, from Panuccio and co-workers in epilepsy. And what's shown here, basically they're measuring extracellular recordings from the piriform cortex. And you get ictal activity and interictal spikes. And they're fairly large, basically. And what they were doing, basically, by the way, is using 4-AP to stimulate the convulsant drug. And what they did is they basically then inhibited two different systems. So this is the same recording. So basically, this is now at a larger time. So you see just one of these, basically. And when you have 4-AP, you have a lot of action potentials. But then when you inhibit with an MDA, you basically do not get the ictal activity, and then you can inhibit the interictal spikes using basically further inhibition with AMP kainate. Okay, so that's known. So now let me go back to what we did in the lab and how that brings some information to this. So basically in the lab we were interested in the piriform cortex. And we were interested, we had done some work in the past, which by the way was done in synchronized firing in the olfactory bulb. So we'll come back to that probably to talk about that. And what we wanted to find out is in piriform cortex, what happens when the animal is active versus when the animal is passive. And so let me get back to that. So basically drastically different states for monitoring an odor, which you're doing right now. Your nose is working right now, but you're not doing this actively. Yet, you know, if we go to the restaurant, we might actually do this actively. Now, so this is our doggy, this is Lady, and Lady does this actively, basically paying attention to the detection, or passively. She's paying attention to something else, but if there's an interesting odor, it'll come in and she'll pay attention. How do you do this? And we found that this was totally different in piriform cortex. So let me go to that. So basically the question then is, how is this related to odor-induced changes in firing rate in piriform cortex? So what we did is we basically measured from the olfactory cortex, and I'm saying olfactory, but it's olfactory or piriform, it's the same thing. And what we do is we have electrodes in the piriform cortex, and we can record from a subset of cells, and we do a go-no-go task. So basically in this go-no-go task that I'm going to show here, we have the mice, and there's an odor that's coming from the bottom. And if it's an odor that they're interested in, they have to lick for about 2 seconds. And if they lick at least once every half a second, then what happens is they get water from here. They get pretty good about that. When you test them with 2 odors that they don't have any idea which one is going to be the odor that's going to give them the water, very quickly, within about 40, 50 trials, they start doing it correctly. So now, how did we record? We recorded from the piriform cortex, and we recorded from anterior piriform cortex, and that is pretty important. I'm going to show in the past, anterior versus posterior. This cortex is very large in a mouse. Okay, so first let's look at active monitoring of the odor. What was that? So basically, to ask them to monitor actively about an odor, we gave them one of 7 different odors as S+, meaning if you lick to these, you're going to get the water, but if you lick to the S-, to a different odor, you're not going to get water. And very quickly, they stop licking to the S-. Now, for a passive, it's very much the same. Passive basically is every single one of these odors is reinforced no matter what. So no matter what, if they are there and they lick, no matter what the odor is, they're going to get the water. Okay, so basically in active monitoring of 2-odor discrimination, we get rewarded odors, the 5 to 6 odors, the unrewarded odors. So we're basically asking, is the odor rewarded? So I'll first show an example, then I'll go to the data. So basically, this is just an example of 2 different neurons. I'm calling them U1 and U2, and I'm doing active monitoring in this one. They have to pay attention to, is this the odor that I'm going to get water for? And we had a large number of odors giving a change in firing rate. So this was the actual firing rate. So basically, you can see this here. This is the S-, the one that they're not rewarded for. Very few responses here, whereas with the S+, they had a large number of responses. And this is basically the PSTH showing that difference in response. And this is just 2 different odors. Now, what we did is we then took all of the 216 multi-units, and we looked in 216-fold dimension. So basically, we looked in 260-fold dimension and asked, are these different? Is there a Euclidean distance between the S+ response and the S- response? And here, if we just had 2-fold response, it would be very clear. You know, this would be in close to 0, this would be high, and you would find a Euclidean distance from here to there. Well, we found that 216 cells, something very, very much alike. So basically, this is shown here. This is now the Euclidean distance in those 216 units. And what you see is before they responded, there's absolutely no distance between the go and the no-go, between the S+ and the S-. But then you stimulate with the odors, and you get this very large difference. And if you look at a p-value, asking, are these 2 different, the go and no-go, it goes basically way below. That's -20 in the logarithm of p. So very, very significantly different. And this is where they were able to do it. They were able to do it within about 0.25 seconds. So the question is, when does this happen? Does this happen quickly? And it does. If you look at their licking, this is their licking. Initially, they sort of lick at the middle. 0 is no lick, 1 is a lick. And basically, they start licking, and blue and red are exactly the same, the S- and the S+. And then you get the stimulation, and then very quickly they decide, I'm going to lick for the S+, I'm not going to lick for the S-. You compute a p-value, and again, it's very large difference. But the interesting thing is it happens after that response for the neurons. So the neurons are the first to actually show that there's a difference, and then once they figure that out, they give a response. So this is active, it's basically a change in the firing rate. Does that happen in the passive? And the reason we did that is because initially when people were looking at this in olfaction, they were thinking, you know, in passive and in active, you're going to get a change in rate, and you don't. So basically now we go to passive, which is basically every single odor is an S+, you get water no matter what. And what happens here is very interesting. You basically, I'm showing this for single units or multi-units, but only look at, say, the multi-units. If you look at the active, there's a large % of the cells that respond with this change in rate. There's very few that respond to the change in rate on the passive. So totally different. And I'll come back to humans because there's actually some information in humans on that. So how do you get information on the odor? Because obviously you're getting information on the odor. And what happens is fairly interesting. You look at the sniff log responses, and they do carry additional information. So the way we did this is we recorded from their nose to see when they were smelling. And so basically we record the pressure of the nose, and we can record when they smell, so this is shown here. So basically this is pressure as a function of time, and each of these is a sniff. So basically this is when they had the S pluses, they would sniff quite a bit, and then they would decrease their sniff. So if we look at the sniff, what happens right within the sniff? So let me show you. So first I'll show you the active. So this is when they do determine that they need to respond. And this is the nasal pressure in a single sniff. So basically this is just out and in, by the way they go backwards. And this is the S minus, and this is the S plus within that sniff. And there we saw fairly clear differences in there. Now, were those differences that reflected a difference in the rate or not? And the way we did this, by the way, was the following. To be able to figure out whether there was a difference, what we did is we basically, this is now modulated, this is data that I'm making up, 2 neurons. And what I'm saying is, well, there's absolutely no difference. If there's no difference, what I do is basically the following. If there's a large difference, you see the difference here. That makes sense. This neuron responds more than that one. And how can I actually show that? I can show that by the Euclidean distance. The way I do it is I compute the average for these red ones, and I ask for each of these, am I closer to red or am I closer to blue? Now, in this one, which they're exactly the same, about half the time they're going to be closer to red and closer to blue. In this one, they're going to be about 100% close to blue in this case and close to red in that case. Now, let me show you the result. The result is from the active task. What you can see is that basically they're very close to 50%, and then you give them the odor and there's a large difference, and that means that there's a large difference within the sniff, but in the active, if you then randomize your spikes, you still get a large difference. That's understood because we had shown that there was a change in the firing rate. So basically in this case, in active, there's a change in the firing rate and there's a change within the sniff. It's just because there's a total change in the firing rate. What happens when you look at passive sniffing, though? So the animal is not paying attention, they're sniffing, and what we find here, and I'm just going to show it here, is that in this case, there is absolutely no difference non-locked. So basically, looking at the total rate. But there's a large difference if we look right on the sniff. So right on the sniff, there's going to be a change. So basically, you can get information on the odor if you just look around where the animal is sniffing. And we can actually then do this. This is shown for 2 cells, but we can show it for the large number of cells, for the 216 neurons. And this is discrimination success sniff locked. So this is only when you lock to the sniff. And this is discrimination success that's just rate coding. So basically that's randomized. And if they were exactly the same, they would be right in the middle here. And what we see here is basically that during active sniffing, they're right on there, but when they're either S pluses, so go odors during the active sniff, there's some now that are over here on the basically absolutely very little rate coding, but discrimination success. And when the animal is doing it passively, the green one, you see there's a lot of them that will give you discrimination success. So this is really important. So basically the bottom line is the following. When you sniff and you're interested in sniffing the odor, then there's a changing rate. There's an increasing firing rate. And that, of course, can be a problem. This happens in humans as well. This has been fairly well shown in human, in fMRI. Exactly the same thing. You get odor every single time. So in every single trial you get odors, but in some times you're told, pay attention to the odor. In sometimes you're told, well, pay attention to what I'm showing you in front. And what happens is if you look at piriform cortex, there's a large response when you tell them, think about the odor, and there's very little response when you say, don't think about the odor. Now, they did controls, and you do exactly the same sniff. Now, the interesting thing is that it's been shown, so what this is saying is that in piriform, you can get quite a bit of rate change. Now, the really interesting thing is this work from Luna and coworkers where they went to piriform cortex, and what they showed was that fairly anterior in piriform cortex, so this is a pyramidal cell in piriform cortex, you get very little inhibition by the GABAergic neurons. As you go further back, there is a lot of inhibition by the GABAergic neurons. So this anterior part of the piriform cortex, which is the place where other people have seen problems in seizures, is a place where you can have large problems with GABAergic. And it's when the person is paying attention to that activity of the odors. So where there's a change in the firing rate. And so basically what this tells us, and of course this is work that is related to, but not directly working on the seizures, but it does tell us that there's these changes in firing rate, and that it could be that the changes in firing rate are the ones that tell you something about basically that start a seizure. So basically what we say is that the changes in firing rate, they carry information on odor value. Is this an odor that I'm supposed to do something for or not? Or is it beer? And then there's the changes in sniff lock firing by neurons in piriform cortex that carry information on odor identity. And then the changes in rate in piriform neurons take place only when the subject is actively monitoring the odor. And this anterior piriform cortex that has weak GABA inhibition is likely to mediate epileptic seizures, taking place upon active odor monitoring. Thanks a lot.